Hello everyone, this is Dr. Kalpana and you're watching my YouTube channel Pharmacy Lessons Made Easy. The topic for today's video is microbiological assay for antibiotics. Why do we need assaying them? Because we have to know at what concentration this particular antibiotic will be effective against a specific microorganism, right? So to quantitatively correlate the relationship between the growth of microorganism inhibition and the concentration of antibiotic, we actually perform the microbiological assay for them, right? So let's have a detailed overview about the assay, how it is done, what are the different types, and for which antibiotic we have to take which microorganism to study and which method to be followed. Okay, the test organism, then at what temperature, the incubation conditions like the temperature, time, and the pH which are to be followed for the microbiological assay. Let's begin. Why do we do it? So as to measure the relative potency of an antibiotic against a microorganism. That is the main aim why microbiological assay of antibiotic is done. Now, the basic principle is the microbiological assay of an antibiotic is dependent on how much dose of antibiotic you are incorporating. Okay, so the utilization of particular antibiotic effect, that is growth inhibition effect, on suitable microbe growth curve in dose dependent manner under standard conditions of incubation, like what exactly the pH, with what temperature and what time, right? Time means for 24-hour duration, 5 days or 48 hours, what would be the condition of incubation of that particular microbe in the nutrient condition. Okay, the response, that means the inhibition of growth of microorganism, which is the function of antibiotic actually, which we want to quantitate, monitor, right? So that response is dependent on how much concentration of antibiotic we are using in an assay as compared by the standard known concentration. Okay, because a particular antibiotic at a particular concentration will show a known activity that is determined previously prior to the performing the actual assay for the test sample of antibiotic, right? So in nutshell, what we can say is the microbiological assay is based on comparison of inhibition of growth of microorganism by measured concentration of antibiotic under examination with that produced by a known concentration of a standard preparation of antibiotic which is having a known activity, right? So how to do it? As per Indian pharmacopoeia, there are two main methods which are utilized. One is called method A, or cylinder plate method or cup plate method, and method B, that is turbidimetry or tube assay method, right? So let's discuss them one by one. The first method, that is cylinder plate method or method A, is based on the diffusion of antibiotic from a vertical cylinder, okay, through a assay of stock solution and test solution which are specified for each antibiotic, but with some final diluent which is used for the standard preparation. The assay with 5 levels of standard require only one level of unknown concentration, okay, which is assumed equal to the median level of the standard. Okay, that way this type of assay will be carried out, that is type or method A, right? So that would be based on the diffusion of the antibiotic through the solution or the agar medium, okay, through a agar medium which will be solidified form in the petri dish or the plate to an extent such that the growth of added microorganism is prevented entirely in a zone around the cylinder which contains solution of antibiotic. Okay, so that would be method A. So in this particular example, I have shown this with the help of a diagram. This is a petri dish. Okay, in this petri dish we have taken the media, okay, in which the microorganism against which the antibiotic assay has to be done is already mixed in this agar, solidified agar. Once this is prepared, you have to make certain holes in it. Okay, this will make a cylinder. Okay, once you remove this particular portion like A, B and C, and due to this antibiotic, it will get diffused through the agar medium. This diffusion of antibiotic across this agar cylinder, which is being created by the creation of the hole, right? You put the antibiotic solution inside the hole that is created in the agar diffusion, right? So that will diffusion means the drug or the antibiotic will be diffused across this agar medium. Okay, so as it will get diffused across, so what will happen, the antibiotic, if has a strong potential against the microorganism which is already present in the agar, it will not allow the microbe to grow. Okay, so that would be reflected by this clear region. Okay, this B, C, right? So this one, so this clear region is the zone of inhibition. This is a particular zone which is actually devoid of growth of microorganism because of the presence of antibiotic, right? So this is called zone of inhibition, and this A, B, C, they are antibiotic solutions which are actually created by creating pores in the agar media, solidified agar media inside the petri dish, and inside these pores which have been created, okay, the antibiotic solution have been added. So once as the antibiotic solution will get diffused across this agar layer, the growth of the microorganism will be prevented. And using a particular scale as shown here, using this kind of scale, the zone of inhibition will be noted. Okay, once you have this zone of inhibition, then the assay will be designed in such a way that mathematical model based potency equation can be proved valid. If parallel line mode is chosen, the two log dose response lines of the preparation under examination and the standard preparation should be parallel, and they should rectilinear over the range of doses which are used in the calculation. These conditions should be verified by validity test for given probability. Other than this, other models like slope ratio method may also be used, provided the proof of validity should be demonstrated. Okay, if we talk about the media for this cylinder plate method, the ingredients of the media are peptone, pancreatic digest of casein, yeast extract, beef extract, dextrose, pepaic digest of soybean, that is added in a few types only, agar, then glycerine, polysorbate 80, that is also added in one or two medias, not more than that. Sodium chloride, sodium hydrogen phosphate, sodium dihydrogen phosphate, and finally the pH would be adjusted, right? So based on the quantities, their quantities of different ingredients, as per 1000 ml, different amounts have to be added, and after boiling the media to dissolve the agar, okay, the heated or heating or the boiling of the media has to be done. Okay, so based on this, as per pharmacopoeia, different medium like medium A, B, C, D, E, F, G, H, I and J, they are prepared and will be set at a particular pH value. Okay, so that pH value varies from like media A have a pH, final pH after sterilization 6.5 to 6.6, whereas the maximum pH value is 8.0 for both E and F. Media E and F, it varies from media D and E, and it varies from 7.8 to 8.0 for both of them. So that way, for different types of media, different value of pH is to be adjusted. Then standard preparation will be prepared for an antibiotic, which is actually you have to take an authentic sample of antibiotic for which the potency has been precisely determined with respect to a reference of international standard, right? So that would be expressed in international units or in microgram per mg of the pure antibiotic. So this standard preparation for India is actually certified by Indian Pharmacopoeia Commission, that is IPC, situated in Ghaziabad, or by any other notified laboratories, and that should be maintained properly. This standard preparation may be replaced by a working standard which is prepared in any lab, and should be comparable or compared at definite intervals under varying conditions with the standards. If we talk about buffer solutions, they are actually prepared by dissolving different amounts of disodium hydrogen phosphate, potassium dihydrogen phosphate in sufficient water to produce 1000 ml after sterilization, with adjusting pH using phosphoric acid 8 molar solution. Okay, and the final pH which is to be adjusted will vary from like 4.5, buffer number 3 whose pH varies from 4.5 plus minus 0.01 to maximum value of 10.5 plus minus 0.1 for buffer number 5. So there are 6 buffers which have been prescribed by Indian Pharmacopoeia: buffer number 1, 2, 3, 4, 5, 6 and 7. Now, to prepare a stock solution, the standard preparation of given antibiotic, accurately weighed and previously dried, the solvent specified as per the table that I'll show you later on. It would be like this table. So in which the antibiotic name is given, okay, which assay method is to be followed for which antibiotic, whether that particular sample has to be dried previously, and the initial solvent in which the antibiotic solution has to be prepared. Like for amikacin, the initial solvent for further diluent is water, and the final concentration which is to be used is 1 mg, and before 14 days it has to be used. Okay, the same way if we talk about any other antibiotic like nystatin, for it also the assay method A has to be used. It has to be dried prior to use. The stock solution has to be prepared using DMF as a solvent, and the final stock concentration per ml is 1000 units. It has to be utilized on the same day once it is prepared, right? The day it is prepared. So that way the standard solution has to be prepared, okay, on the day of assay from the stock solution. This standard solution has to be prepared by successive solutions, increasing stepwise in concentration, usually in the ratio 1:1.25. Okay, for the method A, or later on for method B also the same method has same procedure has to be followed. So this final diluent which is specified in the table has to be utilized in a sequence in such a way that the middle or the median concentration is whatever is described in the table 3, that should be equivalent to it. Okay, now if we talk about the preparation of sample solution for this, for the information available for the substance under examination, that we call as unknown sample, we have to assign it as an assumed potency per unit weight or per unit volume. Based on this assumption, you have to prepare on the same day on which you are performing the assay stock solution. The same test solution has to be done as specified in the table, but the same final diluent should be used as you have used in the standard preparation. Okay, the assay with 5 levels of the standard require only one level of the unknown at the concentration assumed equal to the median level of the standard. Okay, so if we talk about test organisms, the test organism for each antibiotic is listed in the Indian Pharmacopoeia, and their identification number is as per American type culture collection, like ATCC it is called, and the culture on the slant has to be maintained on the medium and under the incubation conditions which are also well prescribed by the Indian Pharmacopoeia. For example, the test organism for amikacin is Staphylococcus aureus, and for amphotericin B the test organism is Saccharomyces cerevisiae. Bechitracin, the test organism is Micrococcus luteus. For bleomycin, the test organism is Mycobacterium smegmatis. For carbonicillin, Pseudomonas aeruginosa is used. For chlorotetracycline, Bacillus pumilus is used. For erythromycin, Micrococcus luteus is used. For famicidine, Bacillus pumilus and Bacillus subtilis is used. For gentamicin, Staphylococcus epidermidis is used. For kanamycin sulfate, both Bacillus pumilus and Staphylococcus aureus are used. For neomycin, Staphylococcus epidermidis, and for novobiacin also Staphylococcus epidermidis is used. For nystatin, Saccharomyces cerevisiae, for which amphotericin B also, and for nystatin, both of them Saccharomyces cerevisiae is used. For oxytetracycline, Staphylococcus aureus or Bacillus cereus or macrolides can be used. For polymyxin B, Bordetella bronchoseptica, okay, that may be used. And spiramycin, for spiramycin, Bacillus pumilus is used. For streptomycin, Bacillus subtilis is used, and Klebsiella pneumonia is also used for streptomycin. For tetracycline, Staphylococcus aureus and Bacillus cereus is used. For tylosin, Staphylococcus aureus, and for tobramycin also Staphylococcus aureus is used, right? So that way Indian pharmacopoeia is very well specified the antibiotic as well as a microorganism which is to be used for microbiological assay, right? So this name of antibiotic and the particular microorganism which is used for that particular antibiotic is very important from exam point of view, especially for GPAT exam, because that would be a multiple choice question exam, multiple choice based exam in which you have to choose the correct answer. So that for that particular exam this question is very important, and it might be asked for this particular antibiotic which microbial strain is used for assay, antibiotic assay or microbiological assay. So you have to choose the correct answer. Okay, so what we have studied so far is the what is microbiological assay, okay, and what are the different medias which are used for performing microbiological assays, and the different methods are like first one we have studied so far in detail, cylinder plate or cup plate assay, which is based on the diffusion of antibiotic across the agar medium, and the how to prepare the buffer solutions, the test organisms, the standard solution and the stock solution, right? So that way, moving to the next assay or the method, that is turbidimetry or tube assay method. This method is particularly based on the fluid media which is containing the antibiotic. First it has to be inoculated with the specified microorganism, and then it should be incubated. The growth of microbial culture will result in the turbidity, okay, after incubation. The turbidity which is produced by the test concentration of the antibiotic will be compared with that produced with the standard solution of the antibiotic. That way, by comparing the turbidity produced after the growth of microorganism, it can be assumed and quantitatively correlated that what is at what particular concentration this particular compound or the antibiotic is showing how much of activity. Okay, so the for B method, using several inocula, the procedure as described by the specific antibiotic assay will be run by only the high and low concentration of the standard response curve. After incubation, the absorbance of the appropriate tubes will be read, and determining which inoculum produce the best response between the low and high antibiotic concentration, and that will be used for this inoculum, okay, for this particular assay as an inoculum, right? So if we talk about this table, then particularly concentrating on like which antibiotic, which method. If we talk about amikacin, method B is used. For amphotericin B, method A is used. Bacitracin, method A is used. Bleomycin, method A is used. carbonicillin also method A, chlorotetracycline method A, as well as B for erythromycin method A, famicidine method A, gentamicin method A, kanamycin sulfate method A and B, neomycin method A, novobiacin A, nystatin A, oxytetracycline A, as well as B, polymyxin B method A, spiramycin, streptomycin method A. For streptomycin method B is also followed. For tetracycline method A and B, and for tobramycin and tylosin method B is used under different conditions of solvent, final concentration, and they have to be used at what day, like on the same day amphotericin B and bechitracin has to be utilized, on the same day nystatin has to be utilized, on the same day tylosin has to be utilized, or there are conditions like number of days are 14 also in some of the cases. It may be 4 days, it may be 30 days depending on the concentration, on the nature of that particular antibiotic. And the test dilution, the final diluent, is specified by Indian Pharmacopoeia for each and every antibiotic, right? So that way it has to be carried out. So after performing both the test, antibiotic assay, test A as well as test B, you can easily carry out the microbiological assay and quantitatively say which particular antibiotic is having activity against any particular microorganism or not. If yes, then up to what extent it can be used, what is a particular dose which is effective against that particular microorganism. I hope you people have gone through it in a proper manner and will be able to understand the concept. If you have any doubt, you please write in the comment section. I shall be happy to answer your queries. Keep subscribing my channel. Do click on the like button, and if you click on this notification bell, you will get the notifications once I upload a new video. Thank you. Have a nice day.